to give a glimpse on the progress, some of the progress of the field in the domain of neuroengineering. So at the interface, literally, with biological material, nerve cells in particular. I think I have no remote control, so telepathically, I had it before, but the title is indeed looking at neuron-electrode interfacing. In the next slide, thank you, thanks. So here it's just a couple of days ago, Elon Musk gave an additional announcement that the first human implanted with a brain-machine interface was able to control a cursor on a screen. And this is a composition of an electronic circuit, Bluetooth interface, and an array of several microwires. It's very interesting, but it's not at all new. It has been about 17 years since the start of the first clinical trial for implant in patient to restoring in a tetraplegic patient to restore some sort of motor control. But regardless of this, it's very interesting, very timely for the field of neuroengineering and neuroprosthetics that aims at replacing, restoring, or mitigating, suppressing some symptoms of brain dysfunctions, that progress in both nanoelectronics, microelectronics, and advances in interfacing are happening, are nowadays a reality. I think later today we will have talks for retinal prosthetics, so enhancing or replacing sensory functions, and we are all aware of or familiar with cochlear implants. And indeed, tetraplegic patients controlling cursor, or in this case, robotic arms, are definitely not new. Something that was interesting, however, in the Neuralink technology is this boosting of some sort of novel way to implant electrodes, or this kind of set of wires that are very soft. They are compliant with the stiffness of the brain, presumably reducing the negative effect of the so-called foreign body response, a rejection reaction from the tissue. But ultimately, this shares basically everything in common to the very common concept of extracellular recording. So this is an example of the silicon probes and more fancy neuropixels. There has been definitely significant advances in terms of materials, in terms of fabrication, microfabrication, as well as in electrode counts, multiplexing, and as I said, overall number of counts, but still the interfacing is extracellular, which is the most understood, but yet very primitive. Would be as if in this room we would all be starting talking, and I would have just one microphone capturing the roar of the audience instead of listening to individual voices. We know very well that the extracellular coupling of electrodes is bad in terms of signal-to-noise ratio. So normally nerve signals, so signals from nerve cells, from neurons, is characterized by nerve pulses, but what matters is what leads to the ignition of these pulses, so the integration of the signals, the so-called subtracial activity. And we know well, even in terms of modeling, that when you have electrodes that are outside and they are capturing electric fields that are endogenously generated by neurons through changes of the transmembrane potential, but they are still detecting extracellularly with respect to an extracellular ground or reference electrode, what you actually get is just very strong peaks, the spikes, the actual potential. You completely lose the subtracial activity, which, as I said, is capturing, or is it bearing the information about synaptic inputs and the integration of these synaptic inputs. There are other aspects, such as the endogenous oscillatory effects, for instance, of neurons, that is completely gone, is lost if you only look at supratracial activity like it's normally done. This is an example that has been motivating some of the interest of our lab over the years. Is a recording, very challenging recording from an anesthetized rat done by João Couto, that at that time was a PhD student in the lab in Belgium, now is in Los Angeles at UCLA. He managed to record intracellularly from a layer 5 pyramidal cells, as well as extracellularly by means of a silicon probe, more or less located in the same neighborhood. And what you actually see is that definitely you have increased spatial resolution. You can detect activity from multiple sites, but you completely lose the interesting, apparently resembling a random walk, fluctuation of the subthreshold membrane potential. Now, together with other colleagues, we believe that the fact that this cell is integrating, is receiving inputs, and its membrane potential is somehow witnessing this integration, is somehow the holy grail for decoding the activity of a network. You have, just for free, one neuron that is sampling the activity of the network. So perhaps we should record subthreshold activity from few neurons instead of going from thousands of electrodes, from thousands of neurons nearby, from which, however, it's difficult to separate the individual voices of neurons or making sense of what is just the output of the integration. So in a way here, I'm comparing intracellular access to extracellular access. Now, over the years, many years ago, I had the privilege and the opportunity to work and collaborate with two pioneers of the field of combining nanomaterials and nanotechnologies and neuroscience, Laura Ballerini and Maurizio Prato, who were somehow inspired by some similarity of the extracellular matrix, which is what is the space in between cells that is not empty space, and some just purely morphological feature of nanomaterials when they are deposited, in this case, on a glass cover slip in a sort of spaghetti meshwork. In this case, carbon-based material being carbon nanotubes that could implement somehow some of more authentic and mimic the behavior and the environment that neurons are experiencing, so that you don't have neurons, electrolyte and metal electrodes, or maybe more fancy, polyimide, flexible, but it's still very crude, but you have something that is resembling the complexity, geometrical complexity, nanoscale complexity of that space. So this turned out to be very interesting because these carbon nanotubes are, to some extent, depending on their chirality, they can be electrically conductive, or they can even be semiconducting, and it's very interesting. What we actually did with two postdocs at the time was to devise a physical process to take those probes, the commercial probes, these were silicon probes from NeuroNexus, and we could coat them with these spaghetti meshworks of carbon nanotubes, just to make them more friendly for the tissue. But what was very interesting, it was actually found by, as well as in collaboration with the lab of Laura Ballerini, was that not only neurons love to sit on this spaghetti meshwork of carbon nanotubes, but also these are a scanning electron microscopy of neurons that are cultured. So this is not in vivo, it's not implanted, it's just ex vivo developing neurons from rats. And something that was intriguing at the time was looking at the bundles of nanotubes that just spontaneously, randomly, because we don't have yet a way to control the deposition of carbon nanotubes or the growth of carbon nanotubes in a very precise way. But so randomly, you have bundles that are getting in close proximity with neuronal membranes. And as I said, they are conductive, so it's fed my own imagination of some sort of nanoscopic electrodes that could maybe pinch the membrane of a cell, realizing the best of both worlds. Fancy extracellular matrix, mimicking feature, as well as intracellular access. I can tell you, spoiling the surprise, we didn't manage yet. This is instead a transmission electron microscope of a similar condition, where it's a side view, it's a sagittal section in which you see the substrate where bundles are just spontaneously arising. In this case, these are multi-wall nanotubes, and this is the membrane of a cell, and you actually see that it seems that these bundles are indeed pinching, penetrating the membrane. And There has been attempts, very heroic attempts. I don't think that they were follow-up, in which people did think of this idea and mounted a single carbon nanotube on a cantilever of an atomic force microscope device, and they very carefully push these carbon nanotubes through the phospholipid bilayer that made up a cell membrane. Of course, this is impractical in terms of any interface, in terms of any neuroprosthetic device in which you don't want to have an atomic force microscope to use it. But as a concept of a nanoscopic electrode, this is clearly in the air, and it would have immense advantages, particularly not injuring the membrane like normally patch clamp glass pipettes are doing. And we also know that the carbon nanotubes' properties can boost the behavior of metal electrodes. In this case, I think this is a tungsten electrode originally. And in terms of impedance of the electrode, so electroscopic impedance, as well as in the capacitance, so charge injection capacity, you actually have a lowering of the impedance as expected and an increase in the charge injection capability. So if you coat an electrode, not only you make the surface more appealing for neurons, but you make them better, electrically better electrodes in terms of detecting or stimulating, yet I know, extracellularly the activity of cells. So we were intrigued by this, and we wanted to study in a more systematic way. So once more, we resorted going in vitro because of the ease of physicochemical accessibility and control, instead of in vivo, where conditions and the tissue reaction is a problem. So we consider substrate-integrated microelectrode arrays, very standard devices made on glass substrate and obtained by standard microphotolithography since the 1970s. These devices are commercial. And we cultured neurons, cortical as well as hippocampal neurons from the rat. And we previously, just before plating the cells, we deposited carbon nanotubes meshworks in such a way that only half of the array of this sort of fakir bed was coated. And okay, not particularly exciting. We could see this is a cumulative distribution of the amplitude of the neuronal signal extracellularly detected by individual electrodes in the area coated with nanotubes. They were in red. They were boosted. They were larger. So you get better signals because you have better impedance in this way. And this is another way to describe the same thing. This is the representative picture out of 13 of these experiments, 13 microelectrode arrays. And to our surprise, the fact that you put a meshwork of carbon nanotubes did not shortcut the electrodes. This was my fear, because indeed they are electrically conductive. And so if I have several contacts, maybe if I put indiscriminately carbon nanotubes over the entire substrate, I will basically shunt the electrodes. But this was not the case. First, because we could detect signals, and these neuronal signals were not, so they were there, and they were not sub-millisecond or sub-microsecond synchronized, so there was no crosstalk between channels. And here what I'm showing is that even in the raw voltage, the noise that is related to the interface, there was no correlation. The correlation was not across days in vitro, was not different than the electrodes that were not coated. And this is probably due to the fact that despite you have this meshwork, the percolation length is relatively short. So yes, they are electrically conductive, but they don't shortcut electrodes that are 200 or 100 or 200 micrometers far apart. What we found, however, is that when you plate neurons on the top of these carbon nanotubes, something interesting happens. So not only you get stronger signals, but the periodic synchronized events that are occurring spontaneously in vitro, and it's probably a proxy of synaptic development. So these neurons are reconnecting into functional networks, and because of this connectivity, they start getting activated over and over. As a function of time in vitro, 2 days, 6 days, up to 18 days after plating, so days in vitro, the frequency of these synchronized events that we called burst was increased. It's more 150% increase. So in red, you actually see those MEAs coated with nanotubes showed a systematic higher synchronization. And this has to do with some modification here quantified in terms of the spike trigger cross-correlogram, which is a measure of correlation between spiking activity across different neurons, different electrodes. And here you see the cross-correlation distribution, the cumulative distribution. As the time goes by, you have the red, so meaning the spiking activity detected by electrodes that are coated with nanotubes moves to the right. So it seems that they are getting a little bit more connected. We believe that this is not happening pathologically. This is an interesting feature going even beyond, with the idea that maybe in the future, in 20 years, you would not have only a Neuralink probe in which you have electrodes and the tissue. You will have cells, neurons, perhaps organoids, as the hybrid interface. And those neurons that are growing in close proximity, they have somehow some boosted connectivity. They will do the talk. This is perhaps an ambitious dream. I'm not entering into the details with other work done with Laura Ballerini using graphene, another carbon-based material, where we observe that is considered for novel electronics, that is also resulted in a boosting, in an enhancement for single-layer graphene of synaptic activity, synaptic release, and network activity. So overall, the idea is increasing the signal-to-noise ratio for accessing the subthreshold activity. And for the moment, I didn't give you any result for that. And it's actually turned out to be very, very challenging. And one idea, this is an electrical equivalent circuit model of the coupling between a neuron membrane and a microelectrodes, is to make the seal resistance, which is a measure on how good is the coupling, proximity, or more accurately, the shunting to ground of signals, to make this very high. This would make the signal-to-noise ratio much larger, which is what happens in a classic technique of whole-cell patch clamp, in which here the seal between the pipette and the membrane is very high in terms of gigaohm high. And this clearly, okay, of course, here is the intracellular axis, but this makes possible to have to record also synaptic activity. And here, this is a picture from a review from another pioneer in the field, Mika Spira, with whom we had a project that I coordinated. And he actually envisioned a different approach in which you may not have an extracellular electrode, or maybe you do have an extracellular electrode that has some sort of three-dimensional feature. And maybe instead of passing very small current to burn the membrane, to electroporate the membrane, to cause holes in the membrane, like in this case where these, say, silicon nanowires, they are penetrating the membrane and indeed getting a continuity between intracellular and the electrode itself, he envisioned a way in which the neurons, the neuronal membrane could engulf, could eat, could swallow an electrode just by itself. And this embrace, this engulfment, could be so tight that these input resistance could go very high. He actually proved it, that this was possible mostly in invertebrate cells, in aplysia neurons. Here you see a transmission electron microscope of a mushroom-shaped gold electrode embedded, engulfed, embraced in a membrane of an aplysia neurons. And the shape of a mushroom is somehow inspired by what biology is doing in terms of synapses, where the dendritic spines, the postsynaptic part of the synapse, in some synapses, in excitatory synapses, is resembling a small mushroom. So in this project that I mentioned, funded by the European Commission, together with people from NMI, Peter Jones and Anastasia Moskaliuk, we could find a process, a technological process, to obtain routinely and with very high standard microelectrode arrays that had this three-dimensional mushroom-like protruding electrodes. And the bet was whether these could also work with vertebrates neurons, with mammalian neurons, with rodents. And in the project, we also had the possibility to go to primates. And the main problem is that having 1 micrometer small electrodes made their electrical impedance very high. It was gigaohm high. And this clearly required some trick, particularly coating with P dot or P dot carbon nanotubes. And this indeed worked. Just very briefly, I'm showing a confocal imaging where you see in black these mushrooms and in green the cell. And if you use this stack of confocal images to see, although it's not as conclusive as electron microscopy, but at least it is live, it doesn't require to fix the tissue, you see that from either side, it seems that there is a tight contact between the membranes and the engulfed microelectrodes. You see here a little bit from the scanning electron microscope in which it seems in one case that really neurons, dendrites and neurites are hugging, or the soma itself is actually apparently hugging the cells. So very high impedance can be restored to some level of conventional electrodes if you use P dot or P dot nanotubes. And what we found is that throughout the days, indeed, the number of electrodes that had positive deflections, I'm just going a bit fast here, compared to control electrodes was higher. And the positive deflection could have several explanations, of course, but we thought that this could be an indication that indeed we were somehow approaching this configuration envisaged by Mika Spira of having an in-cell, almost quasi-intracellular coupling. What was, however, very interesting is that just for free, while there was no change or very little change in terms of comparison between the control and these ultra micro electrode protruding in terms of how many electrodes are active, are recording activity, how many spikes per electrodes are detected, is in this graph where for control condition, one electrode, like one microphone, is able to record several shapes, several units, and so it requires some so-called spike sorting to make sense and potentially to separate the sources. But with the protruding, and it could be maybe obvious, could be trivial because they are so small, there were a large majority only recording from a single unit. So maybe the idea or the problem of spike sorting, which is crucial for neuroprosthetics, particularly for prosthetics for restoring movements and reading out signals, reading out thoughts and intention from patient, could be just solved technologically by looking at these novel ways to interface electrodes with a material. Just for the sake of completeness, a few years ago, the recording access to subthreshold activity was achieved, at least transiently, by this group of researchers that, however, did required electroporation. This was a fantastic work, yet requiring to pass and to damage the membrane, perhaps irreversibly, to open holes in the membrane. So concluding, there has been significant advances in microfabrication, as well as something that I did not mention in decoding, making sense, classifying spike patterns. Yet they mostly rely on conventional coupling, extracellular coupling. This has several problems that could be attacked, improved by novel means, novel materials, and novel tricks that are maybe exploiting cell biological properties, compensating for foreign body response and therefore inflammation gliosis. The fact that both recording and stimulation is non-selective. If you have one neuron that is hugging you, well, you're probably going to only hear about his voice, like it has been mostly suggested in the preliminary experiments that I recorded. And so also solving the problem of having discontinuous recording across days. Once more, if you have a very intimate contacts, you get better properties. And I just wanted to give you a glimpse of some attempts in this direction, which I believe is promising, using nanomaterials as well as three-dimensional protruding electrodes. In a way, just as a concluding remark, I think that we are in a very exciting time where looking at the book in which the neural code is written, we have now glasses, but we need the possibility of distinguishing individual letters. We are at that time, and for this we need better interfaces, smart interfaces. Thanks for your attention.